afternoon, everyone. This is a very, timely talk, going back to back with the other theories about ribosome evolution. So I'm Jessica Bowman. I work in Lauren Williams' lab at Georgia Tech. I'm part of the Cool Center. You might recognize this picture from a few minutes ago. So what I'm going to talk to you today about is excavating the root of the tree of life. This is a tree of life which is Gillian Banfield's. Where's the laser pointer?
We got it. Oh, wait, no, that's a different one. We'll give you a light one. That's at least there.
Great. So this is Gillian Banfield's new tree of life. This is based on the 16 different ribosomal protein sequences, consensus of these. You can see there's a lot of diversity here in bacteria. We also have a lot of diversity, or not quite as much, in archaea and eukarya. And if we arbitrarily place LUCA here, so we're not rooting the tree of life, we're just arbitrarily placing LUCA here, what we can do is look back and see if we can excavate this tree of life and ask the question whether the diversity of modern life, what the diversity of modern life tells us about the evolution of biology before LUCA. So as many of you know, this is the ribosome. We have a large subunit. We have a small subunit. The small subunit is responsible for decoding. The large subunit contains the peptidyl-transferase center, which is the region of the ribosome in which the amino-acylated tRNA transfers the amino acid to the growing polypeptide chain. This is a ribosome structure by Harry Knoller's group. We have the small subunit ribosomal RNA in blue, the large subunit in gray. There is a red tRNA here, and this is the PSART tRNA. We know because there's an alpha helical structure, the growing polypeptide chain extending through this long exit tunnel. And so the useful thing for us is that we now have many different ribosome structures. So we have ribosomes from all three trees of life, so all three major branches, and then some either crystal structures or cryo-EM structures in the case of some of the more advanced eukaryotes. So if we look at the E. coli structure of ribosomal RNA, this is a piece from the large subunit near the peptidyl-transferase center, what we can see is that we have an RNA backbone here indicated in red. This is of E. coli bacteria. If we superimpose this with a Haloarcula maris mortui representing an archaean, so from another domain of life, and superimpose this with a yeast from the eukarya, and then with Homo sapiens, a mitochondrial ribosomal RNA, the same region of the large subunit, what we see is that over 3.8 to 4 billion years, this ribosome really has not changed, which is kind of amazing. We can find the boundaries of this conservation, this conserved core, as we call it. We can resolve these boundaries within the ribosomes of E. coli, Pyrococcus furiosus, which is another archaean, and also within yeast. Down here, we have the small subunit shown on the left, the large subunit on the right. The blue represents the common core of the ribosomal RNA for the large subunit. The red here represents the common core of the small subunit. If you notice, if you look down this slide, you begin to see that the gray portion, which is the non-common core, or the RNA that's specific to that species, seems to be increasing in proportion to the total amount of RNA. And you would be right in that. If you look at Homo sapiens ribosomal RNA, you can see here, this is the secondary structure. In gray is what we call the common core. In green, these are expansion segments, so the pieces of RNA that protrude beyond that common core. And in pink here is one expansion segment in particular that I'm going to highlight. So this is what we call ES7. If we look at these expansion segments relative to the diameter of the sphere, this common core, and then what we call the eukaryotic shell, so the expansions beyond that common core that's universal to all organisms, we see that these expansion segments, especially expansion segment 7 and 27 of the human ribosome, extend quite a long ways. If we follow this expansion segment 7, which actually begins as a helix 25 in the E. coli ribosome. You notice if we go to yeast here, this purple is this helix 25. It's growing as we then move into fruit fly and then into Homo sapiens. If we look at this in 3 dimensions, and someone emphasized earlier the importance of looking at 3-dimensional structures instead of just sequences or 2-dimensional structures, we see helix 25. This is from the E. coli, the bacteria. We can superimpose that with the crystal structure data of the archaea, or the Haloarculus maris mortui. And what we see is that there appears to be a common ancestor of archaea in bacteria, possibly. We can superimpose the archaea with yeast and see that the ancestor of the yeast ribosomal RNA seems to be best approximated by an archaean. And we can continue on with fruit fly and up to human. This is quite a substantial elaboration on this 3-dimensional structure to have begun with a helix that is conserved in bacteria today. So what can this comparison of modern organisms tell us about the ribosome before the last universal common ancestor? So if we look closely at each of these sites of expansion, in each of these expansion segments, we see that there is a unique geometric fingerprint in most of them, which we call an insertion site. If you notice these two, I believe this is archaea and E. coli, superimposed, and this would be the eukaryotic expansion. So this is kind of like the tree, the trunk of a tree, and a branch growing out of the tree. So this ribosomal RNA grows without perturbing the common core. This is helix 52, so this is a portion of the ribosomal RNA that is different from the one I just showed you, and this is helix 38, which is an entirely different expansion segment. So we can make 2 observations. One is that the modern ribosome post-LUCA grew and is growing by accretion, and that the second observation is that the modern ribosome post-LUCA left some recognizable molecular fingerprints. So if we then combine those 2 with assumptions that the common core also grew by accretion, and in the common core also left, and in the common core, these growth events or fingerprints were also left, and we can go in and look at the common core of the ribosome for these insertion events and actually see back beyond the emergence of the last universal common ancestor. This is a model, and it gives us a testable model. So this is the superimposed helix 24. So this is actually in what we call the common core, the ancestral expansion. And here's a branch of ribosomal RNA coming out. So this would be some kind of progression in LUCA, or before LUCA. And we also see this type of insertion site in tRNA, amazingly. So this figure up here is entirely attributable to Anton Petrov in the back, as is most of this presentation. But if we look back to this common core, and this is the common core of the large subunit here, we can actually find these insertion sites all through the ribosome, through the large subunit, and each of these color transitions demarcates in a different insertion event in the last universal common ancestor. And these can be integrated through a progression over time and to generate phases, a model of phases of large subunit growth, in which we have this phase 1 and phase 2, a dark blue and light blue. This would be the peptidyl-transferase center here. One thing that I wanted to emphasize is the effect of this model on the exit tunnel and the progression or folding, or the learning that proteins had to go through to fold at some point during evolution. So this is the ribosome tRNA here in blue. We have a growing peptide moving through the exit tunnel. If we look at just the phase 1, so what we think is the oldest part of the large subunit, which is shown here, and we progress through time, consistent with our accretion model. We add phase 2 ribosomal RNA. This develops an exit pore, which would be just the beginnings of this exit tunnel. We add phase 3 phase 4, phase 5, and phase 6. What we see is that the most significant impact of this, the phases and evolution of this in this model had to do with the elaboration and extension of the exit tunnel. So there's another person on our group, Nick Kovacs, who looked at protein folding over the different phases of the ribosome and found that the proteins, so we don't have any ribosomal proteins in phase 1 or 2, but in phase 3, the proteins in phase 3 are really characterized by these random coils. And so this is unstructured protein, and this has been shown by another graduate student from our group, Catherine Lanier. And then phase 4, we start to have some of these beta hairpins. And then in phase 5, we have more complex folds with these beta hairpins and some alpha helices. And so what we think is that the evolution of protein folding is concerted with the evolution of the exit tunnel. I actually need to credit Moran Brinkle-Pinter with some interesting insights into this. So what we think is that when the ribosomal RNA was in the early phases, so it had only an exit pore, no exit tunnel, in phase 3 would have emerged random coil peptides, and as this exit tunnel became longer and longer, we were able to accommodate these more advanced proteins. So the idea is that when the exit tunnel is too short, we get the formation of beta structures. So there's more likely to be aggregation, that sort of thing. So my conclusions today are that ribosomes evolved by accretion of and without perturbation of a 4 billion-year-old common core, and that the evolution of the tunnel and protein folding were likely coupled. I'd like to thank my group at Georgia Tech, especially Lauren Williams, the Cool Center, and our sponsors, NASA and NSF. And here is Anton, our very important person in this work, and also Nick Kovacs is here, and Moran, and also Peter is here, right here. All right, thank you very much.
So we have time for a couple of questions.
So what's the difference between a Drosophila and a human in the elaborations that you're talking about? Surely those are all regulatory functions that have nothing to do with the exit.
Drosophila and human, I think at the time of Drosophila, we probably would have had the more complex protein structures, so the exit tunnel would have been more closer to what we have today. Does that address your question?
I'm just wondering, I mean, it seems like a huge change between those two species, right? So there's got to be some kind of regulation within those species that is regulating the ribosome somehow.
Something beyond the ribosome. Yeah, probably.
Let me just comment on that. The ribosomes throughout the 3 domains have all these kinds of weird insertions and deletions. We do not know the functional aspects of those additions and deletions yet.
All right, great. It was really interesting. One of the peculiar aspects of the tree of life is that there's these really long empty branches, right? And so your model seems to suggest, and I'm not saying it's not accurate, but seems to suggest that somehow the bacterial ribosome sort of stayed constant, right? And could form the node upon which the rest of the long, for the eukaryotes and archaea, which, it could be, but that does require a lot of stasis for the bacterial ribosome, which isn't reflected in the rest of the phylogeny.
Yes, I don't know if you noticed, but even in our bacterial model, there's kind of a common, even though the E. coli structure, ribosomal RNA approximates what we think is the common core, they're not exactly the same. So I think the E. coli may be a good approximation of an ancestral ribosome, but is not necessarily something that was fixed in time in which eukaryotic ribosomes grew upon.
Yeah, you can speak to it after the seminar because we are already running out of time. So let's all thank Dr. Bowman again.